Hello everyone, I am Atharva, a PhD candidate at Center for Neuroscience, IISc. Today I will be presenting my uh, work review for the past 1 year for my thesis titled Role of Dopamine, Basal Ganglia and Motor Cortex in Reward-Driven Motor Learning. Our motor systems are uh, uh, can generate quite complex skills which are executed in very multidimensional space with a very sometimes very simple goals like beating the batsman if you are a bowler. But our motor system not only generate, they also learn new skills in a, in a by trial and error, where the goal is to accumulate as much success as you can. um, the hallmark of these kind of learning is that with practice over days or months, uh, the period can vary depending on the complexity of skill. You start accumulating more and more reward, and your actions become more and more consistent. The variability reduces. So how do we study this in lab? So we have used rats as a tractable model system, where we establish this behavior task called angle press. In the task, some water deprived animals have to press the joystick at precise angles to get water reward. So in the video coming here, you will see that rat gets rewarded for certain angles but not the others. It goes to drink water at the bottom. So the task structure overall looks like this: that the there is this pink rewarding zone you see, that that entails that any press within this zone is rewarded, other outside this zone is not rewarded, and in next session this rewarding zone can change and the rats have to adapt their movements accordingly. What we find that rats actually have to learn these new angles to improve their reward rate. So typical training period looks like this. Each green line is a session and the targets are in the red. So any trial in the red zone is rewarded, and you can see that the rats learn to follow the rewarding zone. You can summarize this data as a fraction of trials with correct angle, and on x-axis the trials from the start of the session. So within session you can see that the rats start to improve within each session on an average, and this delta performance is a metric that I will use again in this presentation. For uh, enabling us to do long-term studies, we have established this uh, automatized setup, which, which can give us very high throughput behavior results. You can get millions of trial using a few rats. So the question here is that we know that rats can learn, but how does their brain implement this kind of trial and error motor learning? So what I'm showing on the screen is a very basic schematic of our motor system, in which each of the regions play some kind of role in the learning. But what our focus currently will be on this motor cortex basal ganglia loops here, and the field still is evolving, and the conventional roles of motor cortex and basal ganglia have been challenged by recent studies in species like songbird. So the role of them in learning kinematics using a trial and error approach is not clear, and that's what my question is. So my M1 is the investigate the role of motor cortex and basal ganglia in trial and error motor learning. How do we do this? The experiments involve training the rats on the task, so that they become expert. You have the expert data now. That is called as pre-intervention data. Now we can do any kind of intervention and again put back the rats on the task, and we can collect post-intervention data. So what we do is, as an intervention we do silence. We silence the MC or DLS activity, either one of them in a rat, using some excitotoxic agents. We inject them very precisely, and we get such kind of focal lesions out of them. Now I'm showing you the results here, that This is an example animals data where the pre-lesion animal was able to learn. It was following the rewards quite nicely. It's around 40,000 trials worth data. Post lesion, again similar, 30,000 trials. It just don't change its angle at all. The learning rates would have drastically dropped. The animals with motor cortex lesion tell a little different story. For pre-lesion, you can see that they are able to learn. Post lesion also they are able to learn, but can still gauge that there is a more variability in their actions. The reward rates might be little lower, and that is reflected in the kind of fraction correct plots that I have shown you previously. So the pre-lesion and are in the green. For a unilateral lesion are in the salmon color, and the bilateral lesion are in the red color. So you can see that the performance drops drastically. The learning is totally uh, abolished in DLS lesion, but in MC lesion it still shows some kind of learning, but the reward rates are overall low. Once you summarize data for all the animals that I have done till now, you get something like this. That for DLS regions, which are in purple color, for unilateral and bilateral region, the performance is catastrophically lost. MCLOS lesion shows a gradual decrease, and DMS lesion, which serves as a control also, do not show that kind of decrease as we see in other lesions. Of course, I have to accumulate more number of animals in here in this data set. But once we have established, okay, this region is important, the question next comes is that the reinforcement learning requires a kind of reward feedback after generating the movement so that you can again repeat that movement. So the question is, what kind of learning signals or reward feedback, which serves as a learning signal, do these areas, specifically DLS, receive? So the different kinds of learning signals that are popular in RL models, in reinforcement learning model, are simple reward feedback 1 or 0 as the uh, learning signal, or expectation of reward at trial t, like which is a value signal. That is also quite, you can learn through all of these learning signals. Reward prediction error is just the difference between what reward you received and what expectation, the surprise in reward that you received. So what my aim to becomes is the characterizing the dynamics of dopamine in dorsolateral striatum during trial. Why dopamine? Because the it is known to play a significant role in encoding the reward signals. So what we do as experimentally here is that we characterize the DA dynamics in DLS using fiber photometry. So we inject a dopamine indicator inside the brain, and we record using optical fiber. So we inject, we wait for the expression of that indicator, and then we record animal while it is doing the task. So the task has another aspect to it that I skipped previously, that the there is a tone after rewarded presses. As soon as the press is complete, if it is going to reward, there is a tone. For non-rewarded presses, there is no tone. So what we see is that uh, if this is the dopamine plot, it is aligned to 0 is the press start here. Reward Q is present as a white line that you see here, and actual reward is delivered somewhere here. So you see a peak after reward is like after the reward Q. So basically you see something like this. If you segregate trial wise rewarded and unrewarded trial and compress them into a histogram, you see something like this. The Z score dopamine signal starts approximately at 0, and then it increases during the press more for rewarded trials, and then it goes further up in case of a tone. What we find that it shows a typical RP characteristics where if you segregate this rewarded trials based on the reward rate that previous trials had there. So if previous trials were rewarding, that is high reward rate, the expectation on current trial will also be sort of high. So if expectation is high and you get rewarded, the surprise is low. That is in the low is a high reward rate plot here, and the low reward rate plot is higher surprise. So that is, this is a very typical RP kind of characteristic. The relationship becomes opposite in case of unrewarded. So if the history was highly rewarded, then you get no reward in current trial, then you'll see a higher surprise and higher dip. So this is what I showed was a typical modulation of RPE with reward rate or reward history. But I am also interested in if this is also modulated by the current action. Do dopamine system also has information of current action that has been taken? So that we have got some cues that it might be true that we have some catch trials in our behavior where some incorrect press angles are also followed by tone and some correct press angles are not followed by tone. So if you see here that whenever there is an incorrect trial and it is followed by tone, you see a very high increase in dopamine with, which is not seen as correct press angle. And similarly, it's also in the unrewarded case. So what this may mean is that the expectation to calculate RPEs may include the information about current action and reward history. I have to verify this using more analysis tools like regression methods, and it may mean that algorithms like advantage actor critic may explain better than actor critic models. And from our previous lesion study, we can say that DLS may be the actor that learns through dopamine learning signal. This is it for my work report. Thank you. I also like to thank my supervisor Ashesh Dhawale and the lab members and PMRF for funding my research.